All right, so continuing with our brain regions, my recommendation is to go back to the blank Jamboard and fill this out multiple times so that you have practice with it and you just get it ingrained and strengthen your synaptic connections regarding the material. One of the best ways to do it is to go back 90 minutes after you learn something. That's a good, like the sweet spot for timing in repetition. Do that and then do it the next day and then do it 2 days later, and then you will help encode that, and we will learn the specific cellular mechanism for that when we talk about learning and memory coming up in one of the later chapters. So practice it over and over again. Yes, you do have to know the structure names, and yes, you do have to know the function names. So continuing on, getting a little bit more in depth. So we looked at the overall structure of the brain and the outside of it, just by looking on the outside of it. But now what we're going to do is we're going to go take and cut through the larger fissure, the one that separates the two hemispheres, and we're going to look at the things that you can see in the middle of the brain. But before we do that, sorry, I got ahead of myself a little bit, we're going to look at specifically a little bit more in detail in the primary somatosensory cortex. So this one right here. And then also there is a primary motor cortex, which is the number 2, which is the precentral sulcus. The one that's represented here is actually the sensory one, but the motor one is relatively similar. And so what we've done, or what they've done here in this image, is basically they have taken a section of the brain by taking it and cutting it right through this section of the brain. So they've cut it, and this is only half of it, so there would be technically another, here's the ventricle, and then there would be a the other hemisphere, right, would be over here on this side. So this is only one picture of one side of the brain, but just recognize there's one on the other side as well. So the feet and the legs down here, as far as the sensory cortex is located in that interhemispheric fissure, so right in the section that's right in here, right? So down here are going to be really deep down in there. The as best I can draw or write here, the legs and feet are down in there. But then when you get to the surface of it that you can actually see, not buried down in that interhemispheric fissure, what you see is that you have a little bit of space that is for your torso. So that would be basically this section right here. And I'm doing the best by estimating here. So this section would be the torso. Let's see if I can do this just a little bit better. Typing it instead of writing it out. So this little section right here is going to be the torso. But then you have this really large section, and if you think about this little space right here, and this space here, this is twice as big as the entire area of the cortex that is devoted for the torso. That's devoted to the hand. And so the hand is highly represented in the somatosensory cortex. So about this much of it is the hand. So if you take this little bit and multiply it by 2, you get the hand. So a much larger representation in the cortex devoted to the hand as opposed to the torso and even the legs. And then, as you can see, the rest of that gyrus is designated for the face and lips. So the concept here is that we have a larger portion of the cortex, and specifically this somatosensory cortex is devoted to the hands and the face. In comparison to the rest of the body. But if you think about the surface area, just look at your hands and then look at your face and then bring your hands up there to your face, right? And think about how much surface area that is versus the rest of your body. All right, so a small surface area of skin has a larger representation in the somatosensory cortex in the brain than the rest of your body. And the concept behind that is that if you think about how you interact with your world, you use your hands for many things. You're not going to like pick something up with your shoulder or pick something up with your knee, right? So you don't need a lot of detailed sensory input coming in from your thigh as opposed to your hands. When you are working and interacting with the world, you are using your hands to give you more information. And so more of the cortex is devoted to your hands than it is for a lot of the rest of your body. And also if you think about the face and the mouth specifically, what would be really important about the mouth? If you think about development, when we are younger, or babies, right? Everything goes to their mouth. That's like their exploratory organ, is because everything, kids will put anything in their mouth. And that is partly because that is how they are interacting with their world in that way to figure out if things taste good. So eating is really important. And the muscles that are developed for eating are an important part of development, because those muscles that are important for eating are also important for talking. So not only interacting with your world in a sense with your hands, but also interacting with, in a social component, interacting with others is in learning how to speak. And so if babies are watching a person, they are watching their face and they're watching their mouth, because that's how they're learning to communicate and talk to people and tell them their needs. So important developmentally, important for survival because you have to eat, and important for helping the grown-ups around you help you survive. So interaction with the world in terms of how you manipulate objects, and then how you interact with other individuals. So that is why a larger portion of the somatosensory cortex, and I forgot to mention, when I say somatosensory, I basically mean touch perception. So all of those touch receptors, like determining if something is smooth, determining if something is rough, figuring out how much pressure you need to use in order to pick up a cup of water. All of those things are somatosensory. So basically skin, touch information, a little bit of feedback in terms of motor, a little bit sensory motor as well. So that is all in the somatosensory cortex, that postcentral gyrus. Complementary to that, the precentral gyrus, the motor cortex, primary motor cortex, also has a somatosensory, or it's not somatosensory, but also has a map to it as well. So the thing that is represented down here is called a homunculus, or small person, or small man. And so you'll notice that the sensory homunculus has really big lips and really big hands, because this is how big a human would be if the actual representation of the face and hands in the cortex was representative of the size of the actual body parts. And so that's why this has really large hands and really large mouth and looks really funny. The homunculus within the motor cortex is similar, except you'll notice that maybe the hands are a little bit bigger and the mouth area is a little bit bigger and the legs and everything are a little bit more spindly, because the fine motor movements that you need in order to make the motions of your mouth and to vocalize, and then the fine motor movements that you do with your hands are much more detailed than what you might do with your legs, right? You have bigger leg muscles. They're bigger, but they aren't as fine-tuned, so they don't need as much input. It's like, you're going to kick your leg. All right, we need to move that one big muscle. Boom, it's done. Whereas if you're going to pick something up, you have to put those two fingers together and recognize how much force you need to pull back, right? So it's a much more detailed and fine motor coordination when you're talking about hands or the muscles on the face in order to vocalize or even in order to eat. So, big summary is that there is a misrepresentation or a greater representation for the face and hands in the somatosensory cortex and in the motor cortex as well, as compared to the larger areas of the body have a much smaller area representation in the brain. Okay, so now we'll move into the identifying structures in the half brain. And we have a few of the structures, so I left the yellow, green, and blue, and then the orange as well. The new ones that I put in here are the ones that you could not see in the full brain image. So you have some new ones here that you're going to be able to see a little bit more detail because now we've cut through and now we have some more things. So see what you can do. Can you do the ones, the yellow, green, blue, and orange we've already done. So see if you can do kind of a 3D visual acrobatics to see if now you can see it in the half brain. And then the new pink ones, see if you can put those in as well. So take a pause and give yourself a chance to do that. All right, so we're going to start with orienting to figure out where the front and the back and the top and the bottom are. So this right here we're going to learn is, and some of the next ones, this is the optic nerve coming in. It crosses at the optic chiasm and it goes back into the brain as the optic tract. But so that means that the eyeballs are going to be right here. So that means that your nostril is going to be right over here, which rhymes with rostral. So this is the rostral part of the brain. The back is where the occipital lobe and the cerebellum are. So that is the caudal. And then the dorsal, remember, like a dorsal fin is on top. And then the ventral side is more like the chest side down here. So just as orienting. And then if we go with the lobes next, again, doing that F pot, so F, P, O, and now we can't necessarily see the T because it's going to go on the outside of the brain through here, but we actually can see this little nubbin right here is the T. So you have the frontal lobe at the front, the parietal lobe up here, the occipital lobe in the back, and the temporal lobe is the one that is right here. And so that means because the lateral fissure separates the frontal lobe and the temporal lobe, so just going back to this image, actually let's go back to this image. So right, the temporal lobe and the frontal lobe, the lateral fissure coming right up here, so take that, cut part of it off, cut half of it away, and then what you'd be able to see is the other temporal lobe coming through the back there, so in between there is going to be the lateral fissure. So this section this space right here is the lateral fissure, or lateral fissure viewed from this side. And then the bigger ones that we had seen, so the cerebrum is all of this folded stuff, anything that has like the gyri and the sulci, that's part of the cerebrum. So the cerebrum's all this stuff. The cerebellum at the base here is this little cauliflower looking thing. And then the spinal cord and the pons we had seen earlier, again going back to this image. So we saw the spinal cord was right here and the pons is right here. So when we cut into the brain and we cut it in half, we actually cut through the pons and through the spinal cord, which is why you can see this section of the pons right here, which is cut. Spinal cord also gets cut as well. So this right here is the pons. A little smaller so we can see it better. Okay, so now what we need to add on here are a few of these pink structures that we had not identified before. So if you haven't gone through those, pause and give your best guess of where some of these might be. And the ones that I'm going to start with, excuse me, are one in between the pons and the beginning of the spinal cord here. So there is a section right here that is a smaller area, but it is the barrier of or the border between the pons and the spinal cord, and that is the medulla, or some people pronounce it medulla, but I don't know, I can't do that. It just sounds weird to me. So the medulla. And then we had already talked about it before because someone had asked a question about it. The pons, the medulla, and the the pons, the medulla, and then this structure up here are going to make up the brainstem. So if you remember, the other thing that was part of it is the midbrain. Let's see if I can make this without making it tiny. Oh yeah, that's good. Okay. So this right here, let me draw it just for a second. So this right here is the midbrain, and one thing that you'll notice, there are two little bumps on there. These are the superior and inferior colliculi, and they are important in eye movement and sound localization. But for our purposes, we'll call that the midbrain for now, even though there are different structures located within that. And then all three of these, the midbrain, the pons, and the medulla, those are all the brainstem. So I'm going to do it like this, kind of draw it, and you can have it in your notes however you want it. But the medulla, the pons, and the midbrain all comprise the brainstem. There. And when we move up a little bit, we see this section right here. Does anybody remember what ventricle this is? Because there's a space in between the two halves of the brain. So that's that skinny space that's between the two halves of the brains. But there's cerebral spinal fluid in there. That's why this looks shiny, is because when they went to cut this brain in half, the knife went through it, and here all this is still shiny, so the knife actually didn't cut anything. When it goes through that, down through that inner hemispheric fissure, or the space between the two brains, that's actually just space. And so the knife actually doesn't cut anything until it gets to this structure right here that connects the two halves of the brain. So you can see that it's more dull, so it's been cut. And then in here is a ventricle, and then here is a ventricle as well. So there's only like one series of axon tracks here that got cut within this ventricle here. And then we had kind of mentioned before where now you get into the brainstem, and that's where you cut more tissue as well. But here, there's no cutting. Here, there's no cutting. And then in this ventricle space, there's no cutting. So if you remember the ventricles, remember this one is the lateral ventricle. This one is the third ventricle. and then coming through the cerebral aqueduct to the fourth ventricle, and then going out through the central canal there. So just as a reference, that third ventricle is where you're going to have the thalamus and the hypothalamus. So the thalamus is a chunkier egg structure, right? It's more three-dimensional. It's going to go back in through here. So the thalamus actually goes back in farther. That's a big egg-shaped structure in there. So this is the thalamus. Oops. Tried to drag it with the highlighter. All right. So this one right here is the thalamus. And then below the thalamus is the hypothalamus. And I always think of the thalamus and the hypothalamus as a little ice cream cone, right? Where the hypothalamus is the cone itself, and the thalamus is the ice cream on top. And hypo means below, and that is why the hypothalamus is called the hypothalamus, because it is located below the thalamus. So now moving back up, you can see this structure that got cut. So this is the structure that connects the two halves of the brain. This is the corpus callosum. So the corpus callosum is that structure, it's axons crossing to the other side of the brain to communicate between the two halves of the brain. So this whole structure, and there are different sections of the corpus callosum, but I just want you to remember that it's the corpus callosum. So that is the corpus callosum. And then right above that, you can see that this is a gyrus. So a lot of students get these mixed up, but the gyrus has to be a bump, right? The corpus callosum is not a bump. That's just a structure moving through, and it got cut. A gyrus, when you're coming through, doesn't get cut. So the shiny part on top is the cingulate gyrus. So the corpus callosum is the one beneath or underneath, and it is cut because it's axons crossing the other sides of the brain. But the cingulate gyrus is this gyrus right above the corpus callosum, and it does go, it goes a little bit farther here. But it basically mirrors the top of the corpus callosum. So this one is the cingulate gyrus. And so those are all the structures that you need to know on the half brain. And just remember, all the pink ones are the new ones. So I do recommend going back to the fresh one and trying it again and trying it again until you can get everything to match, and then give it another day, try it again, and then give it 2 days, and then try it again. And once you have it after that 2 or 3 days, or if you come back to it one more time to review it for the exam, then you know you will definitely have it. That's a better way to encode it into your long-term memory so that you'll be more successful in at least some of your upper-level courses, or just to remember it if you're not a neuroscience major, just to remember it so you get value out of it. So all it takes is repetition. And actually, part of that repetition can be coming up in the things that we're going to look at next. So this next part is kind of a combination of looking at the developmental structures of the brain and what original developmental structures are going to become the parts of that brain that we just labeled. And then added on to that, we're going to think about what are the functions of the parts of the brain that we just labeled. So starting with mapping what we call the secondary vesicles to the adult brain or the developed brain. When we talked about the developing of the neural tube, we had the neural tube that got developed, right? It's just a straw, and that has a hollow center, and then it does close at the top and at the bottom. But then the cells start to divide at a different rate at different sections of the brain. So that during development, it's going to start as a neural tube that's just a tube. But then because the cells divide at different rates, now you have kind of different structures developing. And so that is why anatomists originally said, okay, well then this is clearly a triangle, and then there's a little dip here, so let's call these three different things. And so they called it the forebrain, the midbrain, and the hindbrain. What you'll also see, people will also call them the prosencephalon, mesencephalon, and rhombencephalon. And I'm giving you that because when we get into the next developmental structures, so these will develop further, and then you get into the secondary, what we call secondary vesicles, and then those are called the telencephalon, diencephalon, mesencephalon, metencephalon, and then the myelencephalon. So I've kind of color-coded that here so that the telencephalon is going to be this red stripe structure. And then the diencephalon is more in the middle, and then that is the area that I've colored in darker red since it's represented here. And then the mesencephalon then develops in really just, it stays the midbrain really. And then the metencephalon, these structures here, and then the myelencephalon, this structure down here. So thinking about the way the brain develops, the telencephalon during development then develops into really all of the lobes. The diencephalon in a couple of the structures in the middle. The midbrain stays the midbrain, doesn't advance too much after that. And then the metencephalon goes a little bit, and then the myelencephalon is relatively small. But the best way to remember the order of these is just to start at the top of the brain and then say tel, di, mes, met, my, encephalon. Tel, di, mes, met, my, encephalon. So say it with me if you can. Tel, di, mes, met, my. Tel, di, mes, met, my. Encephalon. And so just practice that. Sometimes that auditory repetition will be helpful. And I found that even I sometimes, when I go to remember these again, I'm like, okay, wait, how's it go? Tel, di, mes, met, my. Okay, yeah, I got it. So sometimes those auditory cues are going to be helpful for you. So I would recommend repeating that. And so now that we have looked at all of the structures, and now that we are focused on the areas of the brain that developed from that original developmental structure of the telencephalon, what I want to do is now go and look and see if you can predict, based on this drawing, what parts of the brain developed from the developmental telencephalon vesicle structure. So I've gotten it on the next frames here. I've highlighted it, right? This is all developed from the telencephalon. And so if you actually look, right, you can kind of figure out how many structures were developed from each section, right? The telencephalon, a lot of structures developed from it. The diencephalon, only a couple. The mesencephalon, just still stayed the midbrain, and that's it. And then the metencephalon only has two structures, and the myelencephalon just has one. So most of it is in the telencephalon. So if you can, pause the video and then bring some of these over that you can see relate and were developed from that developing telencephalon. So give it a pause and give it a try. And so ones that we saw, right, were the frontal lobe, the parietal lobe. So again, this is more practice for the naming of these things, and the temporal lobe that you can see down here. Another one that we saw was the corpus callosum, and I'm not going to get it in a great spot, but then that cingulate gyrus, that spot right above there. And then there are a couple that we haven't seen yet. So these are ones that we have not seen, and there are a few of them that we'd have to do a deeper dissection in order to see. And some of those, one of those is the hippocampus. I had already mentioned that is located down in the temporal lobe. The amygdala is also located down in that temporal lobe, but still all part of the telencephalon development. And then another one that we cannot see in there is the basal ganglia. So that is a motor circuit located within the internal part of the cortex here. So at this point I want to give you two different options. You can either then go through the diencephalon, mesencephalon, metencephalon, and myelencephalon structure-wise, or what you can do is take a pause and then go and try to complete the functions of these particular ones. Whatever you think would work best for you. If you're like, I don't want to blast all the functions all at once, I'd rather put them in this smaller grouping. And if you're going to do that, then all you would do is you go to our week 5 overview, and then the chapter 7C subdivisions is going to give you a chart that has the primary vesicles. So this is the forebrain, midbrain, hindbrain. The secondary vesicles, these are the tel, di, mes, met, my. You would fill that in. And then in the final brain structure, you would put in your frontal lobe, temporal lobe, parietal lobe, occipital lobe, your hippocampus, amygdala, basal ganglia, and the corpus callosum. And then what you would do is see if you can go through and figure out the function for all of these areas. I will, at the very end, give that to you, but I wanted to give you this as a tool if you wanted to break it up into the telencephalon, the diencephalon structures. If you wanted to pause and do that for the telencephalon right now, you can go and give it a try, and then come back and I'll go over everything at the very end. So those are all the ones that telencephalon. Go do the functions if you want to. And then I'm going to continue, and we're going to go into the diencephalon. Remember, tel, di, mes, met, my. So the diencephalon is a smaller structure, and you might notice it kind of looks like an ice cream cone here. So we might know 2 of those that are in there, and one of those would be the thalamus, the ice cream, and the hypothalamus, the cone. So the thalamus and hypothalamus are developed from the diencephalon, or the developmental structure of the diencephalon. Excuse me. But then there are a couple other ones as well, and they're right down in here. I'm going to go back to that other one because it's a little bit easier to see in the zoomed in version. Do I have a blank one somewhere? Ah! I know I have a blank one in this one. There we go. So you've got the thalamus, you've got the hypothalamus, and then right below that, we will learn coming up when we talk about the hypothalamic regulation of the endocrine system and then the autonomic nervous system. But the hypothalamus is connected and controls many of the hormone release from the brain, and that is because it is connected directly to the pituitary gland. So the pituitary gland would be right down here. You usually can't see it because it gets pulled out of the skull when the brain is extracted, which sounds gross, but it's true. But anyway, so the hypothalamus has a direct connection to the pituitary, and that is involved in hormone release. So the pituitary is derived from the diencephalon as well. And then also this structure right here is where the optic nerve comes in and crosses to the other side of the brain. So it's like this X, and you will probably have seen that or will see that in your sheep brain dissections. That where it crosses is the optic chiasm. So the optic chiasm is part of or developed from the diencephalon as well. So if we go back, oops, wrong one. We'll get there, it'll be fine. Okay, so at the front here, right rostral and ventral to the hypothalamus, you have the pituitary gland and the optic chiasm. So all of these, so these are the four structures that are originating from the diencephalon, that developmentally vesicle structure of the diencephalon. Okay, so it goes tel, di, mes, and the mes is just the midbrain. So that is literally all it is. We already know that one. So I mentioned before that the midbrain is actually separated into different sections. So when I mentioned the two little bumps that are on the back here, those are the superior and inferior colliculi, but I don't really need you to memorize those two things. Those two things lumped together are called the tectum. So I wouldn't make you identify that on an image because it's a little bit more difficult. But then the tegmentum is another structure that is in here, and in any of these, a lot of these are axons moving through. There are some nuclei there, so the functions of these are going to be a little bit broad because there are more things moving through here. But the midbrain is made up of the tectum and the tegmentum. So just a little bit more detail on that one. So then it goes tel, di, mes, met, and the met encephalon are these structures right here. And the ones that we have seen before are the pons, right? So that's this structure right here. So the pons is located there. Someday I will get all of these in the right, there we go. The pons is located right there. And then this one is the cerebellum. So the cerebellum is one of the ones that we've already looked at. And for some reason it is not on this particular one. We'll make it. I think I got rid of all like the big ones. Okay, so the pons and the cerebellum, and there is another one that's located in here. It spans a little bit more, but it is the reticular formation. So within there are axons that are crossing there that are considered the reticular formation. So the pons, the reticular formation, and the cerebellum are all part of the metencephalon. And then if you have been following the myelencephalon, there is only one thing that is left, and that is the medulla. So those are all the structures. And then this last one, I direct you to now go to the handout, see if you can then identify the secondary vesicles. Remember, tel, di, mes, met, my, from top to bottom. And then what are the structures within there? And then see if you can make a guess, your best guess as to the functions of those structures. Something that you already know, so you have like your base understanding of it. And then I'll go ahead and give you the basic functions that I have for it. So a function that you have, I might not have on mine just because I've simplified it. But just thinking about the overall basic function for each of these structures. So pause the video, go to that handout, and give it a try and see what you can do. All right, so hopefully you were able to do this part of it just based on what you had already done. I will give you the preview that you will have to do at least some type of reproduction of this on the exam. So just remembering the original development of it is the neural tube. Then that neural tube differentiates a little bit as the cells divide into the forebrain, the midbrain, and the hindbrain. And then further differentiation gives things a little bit different shape so that that forebrain kind of divides off into the telencephalon and the diencephalon. The midbrain stays the one same thing as the mesencephalon. And then the hindbrain divides off into two different sections, the metencephalon and the myelencephalon. Again, tel, di, mes, met, my. Tel, di, mes, met, my. Encephalon. All right. So then The telencephalon develops into many of the structures in the cerebrum. So basically, the telencephalon is a lot of the cerebrum. The diencephalon are the things that are along the midline. The mesencephalon is the stuff, two sections, the midbrain divided into two different sections. The metencephalon is mainly the pons and the cerebellum, including the reticular formation that isn't as visible as those two. And then the myelencephalon is just the medulla. And so if we think about functions of these, we kind of already did a lot of the lobe functions when we looked at the functions of the cerebral cortex. So the frontal lobe is important for higher order cognition. So personality, logic, reason, decision making. The parietal lobe is important for that somatosensory cortex is located there, so sensation. Localization of things. There's some language processing because Wernicke's area is there, and then also attention. And then the temporal lobe is, and I had memory on the other one just so you would know that the hippocampus was there, but now I've got the hippocampus in that section too. So I just went ahead and put language and auditory processing. The occipital lobe, one of the ones that a lot of people might know already, so that's where vision is processed. The hippocampus is the center for creating new memories. The amygdala is important for fear, also face recognition and emotional processing. Oh, and localization-wise, like the hippocampus is right in front of the amygdala. So if you remember the one of the things that one of the tools that I showed you is that three-dimensional rotating brain thing. So if you go look at the hippocampus and the amygdala, you'll see they're both in that temporal lobe, and the hippocampus is kind of a longer that rides along the part of the lateral ventricle that is in the temporal lobe, and then the amygdala just sits as a compact. So amygdala means almond, right? So about a compact almond-sized structure right at the edge of the hippocampus. And then the basal ganglia is a series of different nuclei that are interconnected, and they are going to help with motor planning, also motor processing and habit formation. So it's when you go to learn something new, like if you're going to learn how to drive, at the beginning, you need to pay a lot of attention to exactly what you're doing, whereas later on, after you have learned it, you don't have to pay as much attention. And that is because it has been that motor processing has been incorporated into the basal ganglia, and now it's more of an automatic process. That is actually why one of the things that when we talk about addiction, the motor movements that are done for an addictive substance are hard to get over because it's a habit within the basal ganglia because you're so used to that by the time the addiction happens. And then corpus callosum we talked about a little bit, where it links the two hemispheres. So it's the axons moving across from the cell body in one cell to communicate, or one side of the brain to the cell body to communicate to a cell body in the other side of the brain. And then, so that's all the telencephalon structures. The diencephalon structures are the thalamus, hypothalamus, pituitary gland, and optic chiasm. So basically, if you think of the diencephalon, think of anything that's close to the third ventricle. So that's the thalamus, the hypothalamus, and then a little bit outside of the brain, the pituitary and the optic chiasm. So the thalamus is the sensory relay station. So all the sensory input comes into the thalamus. I wish it would stop doing that. Comes into the thalamus. I know how to fix it. Hold on a second. I'll make this shorter. So all of the sensory information comes into the thalamus. Don't look at the other ones yet. No. All right, so all that sensory information comes into the thalamus, and the thalamus routes it to where it needs to go. So if you have visual information coming from your retina and the optic nerve and then the optic tract, yeah, then that goes to the thalamus, and then the thalamus routes it back to the occipital cortex. So all that information coming in, the thalamus is the center that is going to ship it out to where it needs to go. Then the hypothalamus is responding to all that sensory information, and then it is helping your body react to all that stuff. So you're getting information from your body processes so you can maintain homeostasis, and then you are getting information from your outside world in order to activate the autonomic nervous system, which is your fight, flight, and rest and digest system. The pituitary gland is directly hooked up to the hypothalamus, and when the hypothalamus responds to all of those factors, then it can cause the release of hormones from the pituitary gland. So the pituitary gland is regulated to the endocrine system, and actually I automatically copied and pasted that. Mistake! So the endocrine system, not the autonomic nervous system. I mean, it does feed back some of the hormones that are released from the pituitary gland, then come back and inhibit the adrenaline that is released from the autonomic nervous system, or the epinephrine, also known as adrenaline. So the pituitary gland hooking up to the hypothalamus to help regulate that homeostasis and respond with hormones. The optic chiasm is just where the optic nerve starts to cross and go to the information goes to the other side of the brain. We'll look at that in more detail when we talk about vision. But then the tectum is part of the midbrain, right? So only 4 things in the diencephalon, 2 things of the mesencephalon, which are the tectum and the tegmentum. The tectum is the inferior and superior colliculi involved in eye movement and sound localization, whereas the tegmentum is involved in movement and actually multiple other things, but I just chose movement because it was easier. When we get into the metencephalon, that's when we have the reticular formation right by the pons and the cerebellum. And so the reticular formation, again, involved in multiple things, but I've chosen to focus on movement and sleep because then everything in the metencephalon you can just write is involved in movement because the pons is involved in movement and so is the cerebellum. So the pons involved in movement, and there are axon tracts going through there that are involved in other basic functions. And then for the cerebellum, it actually coordinates with the basal ganglia and helps with coordination, and motor learning is happening there as well. And then the last one, because this video is getting way too long, is the medulla, and that is basic functions like breathing and heart rate. So these functions, I know I've simplified it a little bit, and in some of the future chapters we will get into more of these, like for the hypothalamus we will focus on the autonomic nervous system in a whole chapter. So we'll get into a little bit more of these. Also, anything that relates to vision, we have a whole chapter on vision. But these are just the basic functions that are good to know going forward, so that when I mention the amygdala or when I mention the occipital cortex, you automatically know what those functions are, and then we can build from that. So yes, you have to memorize this. I put this in chart formation because that's how I more easily process it, but if you want to take it and put it in a different format because you learn better in a different way, you should absolutely do that. So practice it. Once you have it, then repeat it, and then repeat it again, and then repeat it one more time, and then when it comes to take the exam, you will absolutely be ready for the content.